Dear students, I am Dr. Nidhi Sharma, Associate Professor Zoology, Government p.g. College, Jhalawar. Today, I shall teach you about the fine structure of the gene. In these slides, I shall teach you about the introduction of the fine structure of the gene, the history of gene, definition of gene, prokaryotic gene, eukaryotic gene, significance of interons. In the introduction, a gene is a specific sequence of DNA containing genetic information required to make a specific protein. Prokaryotic gene is uninterrupted. In eukaryotic gene, the coding sequence, that is the exon, are separated by non-coding sequences called introns. In complex eukaryotes, introns account for more than 10 times as much DNA as exons. Regarding the history of genes or genetics, the classical principles of genetics were deduced by Gregor Mendel in 1865 on the basis of breeding experiments with pea plant. He assumed that each trait is determined by a pair of inherited factors which are now called gene. Mendel's work was rediscovered in 1900 by Hugo de Vries, Correns, and Shermack. William Johnson coined the term gene to the factors denoted by Gregor Mendel in 1865. William Johnson gave the term gene in 1909. William Batson in 1905 coined the word genetics. In 1972, Walter Fiers and his team determined the sequence of a gene in bacteriophage MS2 coat protein. Richard J. Roberts and Philip Sharp found the gene can be split into segments, making it possible that a single gene might be coding for several proteins. Now, what is a gene? The gene is the functional unit of heredity. Each gene is a segment of DNA that gives rise to a protein product or RNA. A gene may exist, exist in alternative forms called alleles. Chromosomes carry genes. Each chromosome consists of a linear array of genes as shown in this figure. You can see the cell, and inside the cell there is a nucleus which contains chromosome, and the chromosome contains the DNA. The DNA is actually formed of nucleotides, and a portion of the DNA is called a gene, which gives rise to protein. Now we come to the prokaryotic gene structure. Genes are called housekeeping genes or specific genes based on their activity. Now, the structural features of a simple gene, that is, has got small genomes, 0.5 to 10 million base pairs. Prokaryotic genes are collinear with their protein, and they've got a very simple gene structure. The prokaryotic gene can be divided into three parts: A. Coding region, B. Promoter elements, C. Terminal region or terminator. Coding region starts with an initiator codon and ends with termination codon. In a prokaryotic cell, there are no introns. Therefore, the coding region is totally uninterrupted. In prokaryotes, the genes are collinear to its mRNA. Now here you can see the dark uh, black colored region, which is the promoter gene sequence. Then you have the transcribed sequence, which is in blue color, and then you have the terminator sequence. Now when this codes the gene, we have the 5 prime UTR AUG signal, which is the start signal for any promoter gene. Then it is the actual coding protein. Then on the 3 prime end of the DNA, the terminator region is present. When you see the DNA, this is there in the rectangular portion. Now the DNA makes the RNA. Here you have got a small DNA sequence which is shown in blue color, which is the template. And then you have got the green region, which is the promoter area, specifically the AUG binding amino sites. Then you have the orange area, which is the RNA coding sequence. And then again, when the gene has been coded completely, it stops at the terminator region, which is shown by the red area. Now, the DNA will make the mRNA. This process is known as transcription. Here you can see the mRNA chain, which starts from the 5 prime end to the 3 prime end. The brown colored area is the leader area, and at the 3 prime end you've got the trailer area. And in between the orange sequences are the protein coding sequences of the mRNA. Now the mRNA will start translation to protein. Here you can see the purple colored amino acid ring as the polypeptide chain. Now, the promoter elements. The upstream elements from the start of the coding region include promoter sequences. 50 to 100 nucleotides upstream of the start codon, start transcriptional initiation site or start site. Now we have to understand that any nucleotide present on the left is denoted by a negative symbol and the region is called upstream element. Example, -10, -20, -35, etc. The start site is symbolized by +1 and Any sequence to the right of the start is called downstreet elements and numbered as positive, plus 10, plus 35, etc. At minus 10, there is a sequence TATAAT, which is also known as the Pribnow box. At minus 35, another consensus sequence TTGACA is present. These two are the most important promoter elements recognized by transcription factors. Now, typical prokaryotic promoter region has TTGAGA, that is minus 35 region as shown in the figure, TATAAT in the blue rectangle minus 10 region. Then you have the RNA start region, and then from there you have the protein start. Now at minus 65 to minus 60, activator elements are present. Activation of the polymerized DNA polymerase takes place. At minus 200 to minus 1000, enhancer sequences are present. Enhancers these sequences enhance transcription by 100 to 200 fold. Here you can see from minus 200 to minus 65 to minus 60 to minus 35. Here you have the enhancers or activators. On the minus 35 you have the TTGACA region, and then at minus 10 you have the TATAT, the terminal region of the gene. These sequences for the termination of the transcription of messenger RNA. It takes place by row dependent mode or row independent mode. In the next part, I shall tell you about the eukaryotic gene structure.